Very good evening students, this is your Prasanna ma'am from NEET World. Now we are going to discuss the second part of photosynthesis in higher plants, where we are going to discuss about C3, C4 and CAM cycles, C3, C4 and CAM cycles, and photorespiration, and lastly we are going to discuss about factors affecting photosynthesis, Definitely the question from the numericals or match the following or statement based is asked in every year from these cycles, and sometimes the question is also asked from factors affecting photosynthesis as well. We will look into mostly the graph which is included for light intensity factor. Okay. So, yes, if we look into light independent reactions, in the previous class we have learnt all the 3 steps of light dependent reactions, wherein all the 3 steps we have discussed, that is absorption of light in the form of photons, photolysis of water and formation of assimilatory power by discussing non-cyclic photophosphorylation, cyclic photophosphorylation and chemiosmotic hypothesis. Now, here we are going to learn the biosynthetic pathways, biosynthetic phase of photosynthesis, biosynthetic phase which is also called as light independent reactions, calling light independent reactions as dark reactions is a misnomer, because they are not dependent on light directly, but they are dependent on the products of light reaction, that is assimilatory power, which includes ATP and NADPH. They are dependent on these products which are formed as a result of light reaction. They are not directly dependent on light, and they don't take place only in the dark. Okay, they take place in the light and dark. Okay. So, yes, they don't just takes place in the night time, and that is why we are not calling it as dark reaction. If we call also, it is a misnomer. Fine. Now, dark reactions or this light independent reactions will occur in these 3 cycles. One is C3 cycle, C4 cycle and CAM cycles. Of these 3, C3 cycles occur in all photosynthetic plants. C3 cycle occurs in all photosynthetic plants. C4 cycle occurs in dry tropical plants. C4 cycle occurs in dry tropical plants, and CAM cycle occurs in desert plants. If you look into C3 cycle, which is also called as Kelvin cycle because it is named after the scientist Melvin Kelvin, wherein he has worked on C14 radioactive isotope, conducted algal photosynthesis in unicellular green algae, that is chlorella, and discovered that the first stable organic intermediate that is formed is 3-carbon compound, which is 3-phosphoglyceric acid. As the first stable organic intermediate formed is 3-carbon compound, he named it as C3 cycle beta, and after the name of the scientist we are calling it as Kelvin cycle also. Then C4 cycle, which is discovered by Hutch and Slack, so we are calling it as Hutch and Slack pathway. In the C4 cycle, the first stable compound formed is 4-carbon organic compound, that is oxaloacetic acid, which is later converted to malic acid or aspartic acid. This occurs only in the C4 plants which grow in the tropical climates. Best examples are, most of the monocots will conduct C4 cycle only, like sorghum, maize, sugarcane. Just only one dicot, that is an exception, amaranthus, it is also conducting C4 cycle. Then CAM cycle, which is also called as Crassulacean acid metabolism. This is also called as Crassulacean acid metabolism, and the first stable product formed is oxaloacetic acid. This occurs in Crassulacean family members like desert climates, like you know these plants are what ma? Succulent plants, for example cactus, agave, opuntia and aloe. And tropical epiphytes also conduct CAM cycle, that is orchids. C4 cycle and CAM cycle is one and the same, but C4 cycle is operated only, you know, C4 cycle is operated both in the mesophyll cells and bundle sheath cells, whereas CAM cells are operated only in the mesophyll cells. This takes place here in CAM cycle. Carbon dioxide fixation takes place during night time. C4 cycle, carbon dioxide fixation takes place during daytime. We will also see the clear differentiations. As of now, you do remember like which scientists have discovered this, and this cycles takes place in what type of plants, and then what is the first stable organic product in them. Coming to the mechanism of C3 cycle, what we have to learn with respect to objective here. The very first step is carboxylation. Carboxylation is the most crucial step of Kelvin cycle because carbon dioxide fixation takes place. The primary acceptor of carbon dioxide in C3 cycle is the primary acceptor of carbon dioxide in C3 cycle is RUBP, that is ribulose bisphosphate, which is a 5 carbon compound. This is a 5 carbon compound. This 5 carbon combining with 1 carbon of carbon dioxide in the presence of the enzyme Rubisco, which is the world's abundant enzyme Rubisco, ribulose bisphosphate carboxylase oxygenase, that has equal potential to bind with carbon dioxide and oxygen. So, this Rubisco enzyme will help in the interaction of carbon dioxide and RUBP, and it will lead to the formation of first stable organic compound, first stable organic intermediate in C3 cycle, that is 3-phosphoglyceric acid, that is 3-phosphoglyceric acid. 2 molecules of 3-phosphoglyceric acid are reduced. Second step is reduction. They are reduced to 2 molecules of glyceraldehyde 3-phosphate by successive reactions in the reduction. Here, for reducing 3-PGA to 2 molecules of G3P, we need 2 ATP and 2 NADPH for every carbon dioxide. For each carbon dioxide fixation in the reduction process, 2 ATP and 2 NADPH are required. 2 ATP are involved in the activity of phosphorylation. It is involved in the activity of phosphorylation. NADPH is involved in the activity of reduction. It is involved in the activity of reduction. So, yes, after successive steps 2 molecules of G3P are formed. 2 molecules of G3P means 2 molecules of 3 carbon compound, on total it is 6 carbon, right. From this 6 carbon, 1 carbon will always escape the cycle for the formation of glucose. 1 carbon will always escape the cycle for the formation of glucose. The rest 5 carbons are regenerated back as RUBP. This final step is called as regeneration. For regenerating 1 RUBP, 1 ATP is required. 1 ATP is required to regenerate 1 RUBP. So, if the statement is like which is the most crucial step to continue the cycle uninterruptedly, so we have to choose regeneration. If he is just asking which is the most crucial step of Kelvin cycle, then it is carboxylation. Okay. So, yes, 1 cycle of carbon dioxide fixation or 1 cycle of Kelvin is involved in 1 carbon dioxide, 1 molecule of carbon dioxide fixation, but then for the formation of glucose, glucose is 6 carbon, right. From each cycle 1 carbon is leaving the cycle, right. From each cycle 1 carbon is leaving. It means to form 6 carbons, 6 cycles have to take place and 6 carbon dioxides are required. Fine. Now, let us look into the energetics of C3 cycle, energetics of C3 cycle, wherein for fixation of 1 carbon dioxide, 2 ATP in the reduction, 1 ATP in regeneration, on whole 3 ATP are required, and 2 NADPH in the reduction process. For formation of 1 glucose, that is 6 carbon, 6 cycles are required, that is 6 carbon dioxide molecules, and 6 into 3 molecules of ATP, that is 18 ATP, 6 into 2 molecules of NADPH, that is on total 12 NADPH are required for the formation of 1 glucose. This is most important, students, you have to remember. Let us exercise few questionnaire based on the energetics of C3 cycle. How many ATP and NADPH are required to reduce 4 molecules of G3P, 3-phosphoglyceric acid, sorry, 4 molecules of 3-phosphoglyceric acid. In 1 carbon dioxide molecule fixation, of course, 2 molecules of phosphoglyceric acid is reduced. When 2 molecules are reduced, it requires 2 ATP and 2 NADPH, right ma? Okay. For 4 molecules of phosphoglyceric acid, how many ATP and NADPH are required? Of course, 4 ATP and 4 NADPH are required to reduce 4 molecules of phosphoglyceric acid. Second question, amount of assimilatory power required to form 1 sucrose molecule. 1 sucrose molecule mean how many carbon? 12 carbon, 12 carbons. Okay, glucose is 6 carbon, sucrose is 12 carbon. How much of assimilatory power is required, he is asking. Of course, for glucose it is 18 ATP and 12 NADPH, right, for 1 glucose, which is 6 carbon compound. For 1 sucrose, which is 12 carbon, just double the amount of ATP and NADPH, that is 32 ATP and 24 NADPH. Clear? Done? Yes? Next. Yeah. Here if you look into next cycle is C4 cycle, which is activated in the plants which grow in dry tropical areas, tropical climates, mostly monocots. Okay, mostly in monocots. So this C4 plants, first you need to understand the anatomy of leaf in the C4 plants to understand the cycle better. So yes, in C4 plants, bundle sheath cells whichever are surrounding vascular bundles. Vascular bundle is surrounded by bundle sheath cells which do not have any intercellular spaces. This bundle sheath cells will show a wreath-like arrangement, flower with garland-like arrangement. This is called as Kranz anatomy of bundle sheaths which are surrounding the vascular bundles. Here in C4 plants, the very first time carbon dioxide fixation is happening in the mesophyll cells where it conducts C4 cycle, and mesophyll cells will have the chloroplast which has grana in it. Okay, so it is called as granule chloroplast. Bundle sheath cells also have chloroplast, but in this chloroplast only stroma is present, grana is absent. So it is called as agranule chloroplast. Agranule chloroplast in bundle sheath cells, and here in bundle sheath cells C3 cycle takes place. So how many carbon dioxide fixations will occur in C4 plants? How many carbon dioxide fixations beta? 2 carbon dioxide fixation will take place. First carbon dioxide fixation in the mesophyll cells, second carbon dioxide fixation in the bundle sheath cells. In mesophyll cells C4 cycle takes place, in bundle sheath cells C3 cycle takes place. And then if you look into the mechanism of C4 cycle, first from the stomata, carbon dioxide enters into the mesophyll cells, and by the enzyme carbonic anhydrase, carbon dioxide is converted to hydrogen carbonate. Carbon dioxide is converted to what ma? Hydrogen carbonate. This hydrogen carbonate, it The primary acceptor of carbon dioxide in C4 plant is 3 carbon compound, that is phosphoenol pyruvate. This is the primary acceptor. Most important, Nana, you have to remember the primary acceptors of both C3 and C4 cycle. This primary, this phosphoenol pyruvate is fixed by the enzyme. Okay, carbon dioxide along with PEP is fixed by the enzyme peptase, that is phosphoenol pyruvate carboxylase, which will only interact or bind with carbon dioxide. Unlike Rubisco, which has equal potential to bind with carbon dioxide and oxygen, peptase will bind only with carbon dioxide beta. So yes, here after that, you know, first organic intermediate that is formed, 4 carbon compound, that is oxaloacetic acid. Oxaloacetic acid by the enzyme malate dehydrogenase, it is converted to malic acid or ascorbic acid. These are also 4 carbon compounds only. And this malic acid or ascorbic acid, they are sent to bundle sheath cells through plasmodesmatal connections, because bundle sheath cell wall is impermeable or impervious for gaseous exchange. It is impervious for gaseous exchange. So the malic acid or aspartic acid is taken to the bundle sheath cells through plasmodesmatal connections. In the bundle sheath cell, decarboxylation happens, wherein this 4 carbon compound is break to 3 carbon pyruvate and 1 molecule of carbon dioxide. This 3 carbon pyruvate is regenerated back to the mesophyll cells. This is regenerated back to the mesophyll cells by the enzyme pyruvate phosphate dikinase and by utilizing extra 2 molecules of ATP. By utilizing 2 molecules of ATP, this pyruvate is sent to the mesophyll cells by the plasmodesmatal connections. Okay, by utilizing how much energy beta? 2 molecules of ATP. And 1 carbon dioxide which is there in the bundle sheath cells, this will undergo Kelvin cycle utilizing 3 ATP molecules and 2 NADPH molecules. So what molecule or what enzyme is abundant in the stroma of bundle sheath cells? Then your answer should be Rubisco. Okay, bundle sheath cells will have Rubisco, and 1 carbon dioxide is fixed by 3 ATP and 2 NADPH. And C4 cycle will utilize additional 2 ATP to regenerate pyruvate back to PEP in the mesophyll cells. If you look into the bioenergetics, for fixation of 1 carbon dioxide molecules, we will see. For fixation of 1 carbon dioxide molecule, of course 3 ATP and 2 ATP through 2 NADPH. 3 ATP are required, 3 ATP and 2 NADPH are required by Kelvin cycle. But then in regeneration step of C4 cycle, extra 2 ATP is taken, right. So on total, to fix 1 carbon dioxide molecule by C4 cycle, how much ATP is required beta? 5 ATP, and how much NADPH is required? 2 molecules of NADPH. For formation of 1 glucose molecule, 6 cycles are taking place and 6 carbon dioxide molecules have to be fixed. So 5 ATP, 5 into 6 is 30 ATP to fix 1 glucose molecule, to form 1 glucose molecule, and 6 into 2, that is 12 NADPH to form 1 glucose molecule. If we exercise few questionnaire, how much extra ATP is required to fix 6 molecules of ATP? To fix 6 molecules of ATP or to form 1 glucose molecule? Or to form 1 glucose molecule, how much energy is required? How much extra ATP he is asking? Not total energy, he is asking us extra ATP. Extra ATP for 1 carbon dioxide fixation is 2 ATP na beta. It is 2 ATP. To fix 1 glucose molecule, 2 into 6, that is 12 extra ATP are required. How much ATP is required for the formation of sucrose molecule? How much ATP, not extra, how much total ATP is required for the formation of sucrose molecule. Of course, for the formation of one glucose molecule, if it is 30 ATP, for the formation of one sucrose molecule, it will be 60 ATP, right? Double the amount of glucose, okay? It is 60 ATP. Then, next if you see CAM cycle. Before entering into the CAM cycle mechanism, you have to be aware of scotoactive stomata. We have learned, right, CAM cycle takes place in desert plants or succulent plants which has sunken stomata and that opens during night time. That stomata which opens during night time is called as scotoactive stomata. Stomata which opens during night time is called as what, ma? Scotoactive stomata. So, during night time when stomata is open, carbon dioxide is fixed by primary acceptor, that is PEP, and by the enzyme pepcase. It is fixed into 4 carbon compound, that is oxaloacetic acid. This oxaloacetic acid by the enzyme malate dehydrogenase, this is converted to malic acid. Malic acid is stored in the vacuole. Malic acid is stored in the vacuole, and during daytime at night time malic acid is stored in the vacuole. During daytime this malic acid is sent outside to the stroma or the mesophyll cells, okay. In the mesophyll cells it is sent outside to stroma, okay, wherein this malic acid is decarboxylated to 1 molecule of carbon dioxide and 3 carbon PEP. It is decarboxylated to 1 molecule of carbon dioxide and 3 carbon PEP. PEP is of course regenerated by consuming 2 ATP, and this 1 molecule of carbon dioxide will conduct C3 cycle. So, here also 2 carboxylations taking place. First carboxylation or carbon dioxide fixation during night time, that in the mesophyll cells by the enzyme pepcase. During daytime C3 cycle is conducted by the enzyme Rubisco, and that also in the mesophyll cells. Malic acid is stored in the vacuole cells, okay. So, this is what you have to remember with Crassulacean acid metabolism. Everything energetics is same as that of C4 cycle, but here only one cell is involved, that is mesophyll cell, for 2 carbon dioxide fixations, and first carbon dioxide fixation or C4 cycle takes place during the night time, okay, because of what stomata, ma? Scotoactive stomata. This table whichever is given in our NCERT is very much important. You have to analyze it, yeah. So, if you look into cell type in which the Kelvin cycle takes place. Kelvin cycle in C3 plants it takes place in the mesophyll cells. In C4 plants it takes place in the bundle sheath cells. In CAM plants also it takes place in the mesophyll cells. Cell type in which the initial carboxylation reaction takes place. Of course, here in the mesophyll, here also in the mesophyll cells, in CAM plants also it occurs in the mesophyll cells itself. How many cell types does the leaf has for carbon dioxide fixation? Third question. Here only one cell type, here 2, one is mesophyll, one is bundle sheath cell. Here 2 carbon dioxide fixations in CAM plants will take place only in the mesophyll cells. Next, which is the primary carbon dioxide acceptor in C3 plants? Primary carbon dioxide in C3 plants is, carbon dioxide acceptor is RUBP. In C4 plants it is PEP, in C4 plants it is PEP. In CAM plants also it is PEP, phosphoenol pyruvate. Number of carbons in primary carbon dioxide acceptor, RUBP 5 carbon compound, PEP 3 carbon compound, here also it is 3 carbon compound. Next, which of the primary carbon dioxide fixation product? What is the primary carbon dioxide fixation product? Here it is phosphoglyceric acid, here it is oxaloacetic acid, here also it is oxaloacetic acid in CAM plants. Number of carbons in primary carbon dioxide fixation products, here it is 3 carbon, right? Phosphoglyceric acid is 3 carbon, oxaloacetic acid is 4 carbon, here also CAM plants it is 4 carbon compound. Does the plant has any Rubisco? Of course, C3 plants have Rubisco, C4 plants bundle sheath cells have Rubisco. Here also Rubisco is there, but which is activated during daytime. Does plant have pepcase? No, C3 plants doesn't have pepcase. C4 plants will have pepcase and CAM plants also will have pepcase. Next, which cells in the plant has Rubisco? Here in C3 plants in mesophyll cells, C4 plants it is bundle sheath cells, in CAM plants also it is in mesophyll cells. Then comes carbon dioxide fixation rate under high light conditions or high light intensity conditions. Carbon dioxide fixation rate will be high in the C3 plants, it is high. In C4 plants also it is high, CAM plants also it is at high light intensities, it will not take place because it takes place in night time. Whether photorespiration is present in low light intensity, photorespiration is not present in any of these plants. In high light intensity, photorespiration occurs only in C3 plants, not in C4 and CAM plants. Then whether photorespiration would present at low carbon dioxide concentration. In low carbon dioxide concentration, photorespiration is not seen in C3 plants, in C4 plants and CAM plants. At high carbon dioxide concentration, it is seen in C3 plants, not in C4 and CAM. Then temperature optimum of C3 plants, as they are temperate plants, it is of 20 to 25 degrees Celsius. C4 plants it is 30 to 45 degrees Celsius and CAM plants it is above 40 degree Celsius. Next, best examples of C3 plants are like tomato, brinjal and all these plants which are of temperate regions. Best examples of C4 are monocots like sorghum, sugarcane, maize, okay, and CAM plants these are like succulent plants, aloe vera, cactus, these are the best examples of CAM plants. Now comes the concept of photorespiration or C3 cycle. Do remember this happens only in the C3 plants. This happens only in the C3 plants, that also because of the enzyme Rubisco. As we have discussed, Rubisco means what ma? Ribulose bisphosphate carboxylase oxygenase. This has equal potential to bind with carbon dioxide and oxygen, right? So when carbon dioxide concentration is less and when there is more oxygen present in the atmosphere, oxygen when it is binding with RuBP, which is 5 carbon compound, this results in the formation of one molecule of phosphoglycolate, which is 2 carbon compound, and one molecule of phosphoglyceric acid, that is 3 carbon compound, okay. As 2 molecules of PGA are not formed, this cycle continuously reverses, okay. This completely reverses. It includes 3 organelles and so much energy consumption to synthesize 1 mole molecule of PGA, and this is called as a wasteful process. This is called as a wasteful process that occurs in the C3 plants, because this consumes ATP, this consumes NADPH, but it does not release oxygen, rather it releases carbon dioxide. And it travels around 3 organelles to synthesize 1 more molecule of phosphoglyceric acid. Let us look into it more clearly, like what is important for our exam, okay beta. So yes, first RuBP accepting oxygen, this will form 1 molecule of phosphoglycolate, that is 2 carbon compound, and 1 molecule of phosphoglyceric acid, that is 3 carbon compound, okay, 3 carbon compound. This phosphoglycolate will be converted to glycolate, phosphoglycolate will be converted to glycolate, glycolate will be entered to the peroxisome, okay. Second organelle is peroxisome, this includes 3 organelles, no, one is chloroplast, then peroxisome and then mitochondria. Here glyoxylate is converted to glycine, glycine entering into peroxisome is converted to serine, glycine is converted to serine in mitochondria. Serine again enters to peroxisome, converts to hydroxypyruvate and then to glycerate. Glycerate then enters to the chloroplast and then it gets converted to phosphoglyceric acid. Here in mitochondria, at the time of conversion from glycine to serine, carbon dioxide is escaped or released out. Carbon dioxide is released out, and here energy is consumed, and energy is consumed in the conversion of hydroxypyruvate to glycerin and glycerate to phosphoglyceric acid also. So, if you look into the cycle, what you have to remember? You have to remember which molecules are present in what organelle. In chloroplast, glycolate is formed. In peroxisome, glycine is formed. In mitochondria, glycine is converted to serine. Again in peroxisome, hydroxypyruvate is formed from serine, and later glycerate is entered into the chloroplast and it is converting to phosphoglyceric acid. So, this continuous process is involving the consumption of ATP, NADPH, rather it is not releasing oxygen, it is releasing carbon dioxide. It is releasing carbon dioxide, that is why it is a wasteful process. Occurs, photorespiration occurs only in the C3 plants because of the presence of Rubisco in the mesophyll cells. Okay. This happens during high light intensities only. Only in high light intensities and low carbon dioxide concentration this process occurs. Yeah. Now comes the concept of factors affecting photosynthesis. Factors affecting photosynthesis are of two factors. We have two factors that affect photosynthesis. One is external factors, and the other is internal factors. Okay. External factors include light intensity, temperature, carbon dioxide concentration, and water. Okay. And internal factors include chlorophyll content, age, size, number, and orientation of leaves. These are the external and internal factors that affect the process of photosynthesis. So, now if you look into here, photosynthesis is a biochemical process which is controlled by several factors. The scientist who is Blackman is a scientist. In the year 1905, he has given out a factor called as a law called as law of limiting factor, which is many at times asked a question, statement based question also. Law of limiting factor given by Blackman, it says about when a biochemical process is controlled by several factors, that process is affected by the factor which is closest to the minimal value. Okay. When it is affected by several factors, that biochemical process is affected, by the factor, which is closest to the minimal value, which is closest to the or the factor which is closest or nearest to the minimal value. Okay, clear ma? Next, we will look into one by one each factor, we will exercise it. Okay. Most of the students will have confusion about these factors affecting photosynthesis. Nothing to get confused, it is more easy. At low light intensities, do you remember beta? At low light intensities, no factor will affect the photosynthesis. Okay, no factor will affect the photosynthesis at low light intensities. At low light intensity, light intensity, no factor will affect photosynthesis. At higher light intensities, at high light intensity, initially rate of photosynthesis increases. Rate of photosynthesis increases as you increase the light intensity. You can see in this graph wherein x axis light intensity is plotted and y axis rate of photosynthesis is plotted, and gradual increase of rate of photosynthesis you can see as you increase the light intensity. And at 10% of total sunlight, at 10% of total sunlight, the photosynthesis rate will be saturated. The rate of photosynthesis is saturated at 10% of total sunlight. If you increase the light intensity further, if you further increase the light intensity, it will damage the chlorophyll structure. It will damage the chlorophyll structure, thereby disrupting the process of photosynthesis, So, with respect to light intensity, what you have to remember, at low light intensities no effect is seen. At high light intensity, as you increase the light intensity, gradual increase in the rate of photosynthesis is seen. At 10% of total sunlight, complete saturation of rate of photosynthesis is observed, and increase in light intensity, if you further increase the light intensity more than this more than the saturation level, if you increase the light intensity, it will disrupt the chlorophyll structure. Yeah. Second factor is what, carbon dioxide concentration. Carbon dioxide concentration is the most limiting factor. According to Blackman, the factor which is closest to the minimal value will show the effect, right? Carbon dioxide is the most or highest limiting factor for photosynthesis. It is because only 0.03% of carbon dioxide is present in the atmosphere. Okay, atmosphere, only 0.03% of carbon dioxide concentration is present. As it is increased up to 0.05% in high light intensities only, again at low light intensity no effect is seen. Okay, at high light intensity, when carbon dioxide concentration is increased up to 0.05%, increase in rate of photosynthesis is seen. Increase in the rate of photosynthesis is observed up to 0.05%. Further increase will not show any effect, it will get saturated. Further increase will not show any effect, it will get saturated. And saturation point in C3 plants and C4 plants is different. In C3 plants and C4 plants is different. In C4 plants, the saturation point is 360 parts per million. In C4 plants, the carbon dioxide concentration is 360 parts per million in C4 plants, okay, which is less. In C3 plants, it is more, carbon dioxide saturation point is more, that is up to 450 parts per million. You can also measure it as milliliters per liter. Okay. Why the saturation point of C4 plants is less and saturation point of C3 plants is more? In C4 plants it is less because the fixation process is modulated by the enzyme peptidase. Okay. This will bind only with carbon dioxide, so it will saturate fast. Whereas in C3 plants, okay, in C4 plants, as it is fixed, carbon dioxide is fixed by the peptidase, it is binding only to the carbon dioxide, so the saturation point is minimal, that is 360 parts per million. Whereas C3 plants, the carbon dioxide fixation enzyme is Rubisco. Okay, the carbon dioxide fixation enzyme is Rubisco, which has equal potential to bind with carbon dioxide and oxygen. So, in this case the saturation point is more, and this is highly used by greenhouse crops or greenhouse plants. By increasing the level of carbon dioxide concentration, rate of photosynthesis is increased. In greenhouse plants, this process is mostly used, like for example bell pepper, tomato, these greenhouse crops. By increasing the carbon dioxide concentration, we are increasing rate of photosynthesis. So, this is all about carbon dioxide concentration. And third external factor is what ma? Temperature. Of course, we have learned, right, C3 plants are temperate plants. Maximum rate of photosynthesis is seen at the optimal temperature, that is 20 to 25 degree Celsius. In C4 plants, that is dry tropical plants, where the saturation point is from 30 to 45 degrees Celsius. 30 to 45 degrees Celsius, it is the optimal temperature of C4 plants to conduct maximum photosynthesis. This is temperature. 4 is water. Mostly at high water conditions or low water conditions, photosynthesis rate is deprived because the initial first response of plant is stomatal closure. First response of plants is stomatal closure. When stomata is closed, how carbon dioxide enters into the cell? When stomata is closed, how carbon dioxide enters into the cell? That is the reason, both in the turgid and flaccid conditions, in water drought conditions and more high water conditions, in both the conditions the photosynthesis process is affected. Hardly, this is an external factor, only 1% it will affect the process of photosynthesis, because water will never be a limiting factor for most of the plants. When coming to the internal factors that affects the photosynthesis, beta, internal factors that affects photosynthesis, of course the leaves which are broad and large and which are aged, matured leaves, they will conduct the maximum photosynthesis, and plants which has high chlorophyll content, or high chlorophyll number, okay, the plants which has high chlorophyll content or high chlorophyll number, which is directly proportional to the assimilation number, okay, assimilation number. So, high chlorophyll number means high rate of photosynthesis, These are the external and internal factors. With this, we are going to end the chapter, that is photosynthesis in higher plants, wherein definitely in both NEET and EAP set you will get 3 to 4 questions from this chapter. Okay, in coming up sessions we are going to deal with the chapter respiration. So, stay tuned. Thank you.